Hello everyone. With the eukaryotic system being more complex and also the genome size being much larger, uh, before the initiation of or before the unwinding of the DNA itself, there are a series of events that lead to the unwinding of the DNA. And these series of events are carried out by several key molecules, which is what this section is going to focus on. So let us look at what are the learning outcomes of this session. One would definitely be able to comprehend that the eukaryotic genome size being relatively large than the prokaryotes have multiple origins of replication, which can begin replication simultaneously, although it is not necessary that all origins fire at the same time. What has been observed by various studies is that several origins fire at the initial phase of the S phase, which is a part of the cell cycle of eukaryotes, and some origins fire only at the later stage of the S phase. Therefore, there are regulations happening that enable some origins to fire at the initial stage itself and some origins to fire at a later stage. But what is definitely taken care of is the fact that because of the presence of numerous origins of replication, the constraint of the genome size is being taken care of. The second thing that is notable is that in prokaryotic systems, the replicator regions have been highly defined by having several conserved and consensus sequences. While across the different species of eukaryotes, as the eukaryotes become more and more complex, what has been observed is that the origins have become less defined. This could be particularly so because since the genome size is that large, wherever possible, if the DNA can unwind, uh, it is it is going to be considered to be an important facet in completing the DNA replication faster. And yet there is a certain element of semblance, so that you still have the replicator initiator interaction happening is very similar to what is happening in the prokaryotes. The origins have been understood by studies carried out in several yeast systems, especially Saccharomyces cerevisiae and Schizosaccharomyces pombe. And over time, various systems in different metazoans, in humans, etc., also have been studied. And there have been found to be certain AT-rich regions which are found to be in the replicator regions of most of the origins. The eukaryotic DNA is in a condensed form. So it is present as chromatin and so associated with histone proteins, and thereby the accessibility of the origins for the unbinding of the DNA can be something that has to be taken care of by the initiation process or by the replication process. This also is therefore aligned to the fact that activity of the origins can be asynchronous. That is, not all origins need to fire at the same time. There may be some that may fire at different points of time, and that could have a relation with how condensed the DNA is. So these are certain things that we will be able to perceive with the understanding of the key molecules in the process that leads to unwinding of the DNA. So which are the key molecules that are involved in the process of the initiation of replication, or rather the process of the replication itself? You have what is called as the origin replication complex, CDC6, CDT1, MCM27, MCM2 to 7. So it is also a complex. All these four together come together to form what is called as a pre-replication complex, and the pre-replication complex marks the regions in the DNA from where the DNA unwinding can begin. Although it is not necessary that wherever you have a pre-RC formed or a pre-replication complex formed, the DNA is going to unwind at that point. However, the formation of the pre-replication complex is a preparation that happens in the G1 phase before it goes to S phase to mark those regions where the DNA can unbind. So these are certain molecules that we will definitely discuss in this section. There are two regulatory molecules associated with the cell cycle that help in the initiation of replication, and that is the cyclin dependent kinase and the DB4 dependent kinase. Both of these are instrumental in initiating the process of replication. There are CDC45 and SLD. SLDs are initiation factors that are again required at various steps. GINs, DPB11 and MCM10, these again are important for converting the pre-RC, that is the pre-replication complex, to a pre-initiation complex. And the pre-initiation complex basically ensures that unwinding of the DNA happens. What is interesting to note is that CDC45, GINs, DPB11 and MCM10 also are involved in elongation and termination. So before the DNA polymerases actually initiate or add the first nucleotide, all these molecules have an important role to play, and hence this session will basically focus on these molecules. These molecules, that is DNA polymerases, primase, RPA, which is equivalent to the single-stranded binding proteins in the prokaryotes, pCNA, FEN1, DNA ligase, RNase H, DNA topoisomerase, telomerases are more involved in elongation and termination, and hence they will be considered in a different section. Now let us go to origin replication complex. The origin replication complex, which, as mentioned earlier, is responsible for marking the DNA for formation of the pre-replication complex. So in fact, this origin replication complex, which is a heterohexamer, that means it is made up of 6 subunits and all the 6 subunits are different, hence the name heterohexamer. And this heterohexamer is the start point for formation of the pre-replication complex. Now, this origin replication complex has been extensively studied in various species now, but the first species in which it was studied in 1992 was in the yeast, Baker's yeast, which is what is Saccharomyces cerevisiae. So we're going to discuss the assembly of origin replication complex, keeping in mind the Saccharomyces cerevisiae, but one can extend the same assembly pattern to different species across the eukaryotes. So let us understand what happens in the Saccharomyces cerevisiae. When the Saccharomyces cerevisiae has a replicator region which is 100 to 200 base pair long, and within this 100 to 200 base pair long region is an autonomous consensus sequence, which consensus and the sequences are also conserved, and this region is the one where you have the origin replication complex coming and binding. So this is crucial. Studies where this portion has been removed has shown that the ORG replication complex does not assemble or bind to the DNA when this region has been removed. Therefore, this A element, ACS or A element, in which about 17 base pair, from the 17 base pair, 11 base pairs are highly conserved region, with several T's very important in the highly conserved region. And these T's are also highly conserved. So effectively, the T's present, or the 11 base pair that is highly conserved, is responsible or important for the origin replication complex to recognize the replicator region. So therefore, the recognition or the assembly of the ORC at the origin is thanks to these 11 base pair highly conserved region. It also binds to B1 element. So other than the A element, there are present what is called as the B elements. This is B1, B2 and B3. Of the 3 B elements, B1 is again having certain conserved sequences to which certain components of the origin replication complex can bind. The B2 element has basically the initiation site for bidirectional replication. So this is where the unwinding happens. So effectively, A and B1 are required for ORC to assemble, and B2, this region, is what is the DNA unwinding element. So this is where the DNA is going to open up, or this is where the DNA is going to unwind. So effectively what one can observe is that close to B1 and A is the region present where the unwinding of the DNA can happen, which is very similar to what is observed in the ORIC of the E. coli, where you have the consensus conserved 9-mer sequences close to the 13-mer region, and that's the 13-mer region which has an AT-rich region and can unwind. So a similar thing is observed in the Saccharomyces cerevisiae as well. The the B3 element is an accessory element, and it is just about 22 base pair long. And in the Saccharomyces cerevisiae, it has been noted that the B3 element can bind to a transcription factor ABF1. So what is observed is that these replicator regions also help in the process of transcription. Now let us look at the fact that the B2 is required for the pre-RC formation and also for the helicase, that is the MCM27, to come and bind. It is important because when there are mutations of the B2 element or mutations are induced in the B2 element, it has been observed that the pre-RC complex is not formed properly, and thereby the loading of the helicase is also not proper. One important factor that one should keep in mind with respect to the origin replication complex is that its function is coordinated with ATP binding and hydrolysis. So this fact that the components of the ORC are able to bind to ATP and also hydrolyze ATP plays a very important role, not only in the assembly of the ORC but also in the formation of the pre-RC. So when one looks at the individual subunits of the ORC complex, you have ORC1, ORC4 and ORC5. All the three subunits or all the three proteins belong to AA plus ATPase superfamily. And as known in this, there are very distinct conserved domains that enable the proteins belonging to this family to bind to nucleotides. So there is definitely a nucleotide binding domain, and it also has an inherent ATPase activity. Of the three, ORC1 is the largest protein, and this is also the largest in the entire origin replication complex. You also have what is called as ORC2 and ORC3. They are distantly related to the AAA plus ATPase family because it has been found that there are a few domains which are very similar to what is present in ORC1, 4 and 5, but these two lack the conserved region of the ATP binding site. And so that is how these two become structurally different from ORC1, 4 and 5. But nevertheless, there are several domains that are very similar to what are present in these proteins as well. The subunit which is the most different is the ORC6, and it is also the smallest subunit of the entire ORC6. This is not an enzyme. Unlike the others which have the enzymatic activity of ATPase, ORC6 does not have any enzymatic activity for the reason that it does not belong to the AAA plus ATPase family. However, its being part of the ORC is important for making the pre-RC complex, because where there have been studies carried out with mutations of the ORC6 and the ORC6 not being able to assemble with the remaining 5 ORC subunits in the Saccharomyces cerevisiae has shown that the pre-RC complex is not able to form properly, and also they are not able to load the helicase because of the absence of the ORC6. Therefore, this becomes a very important part of the ORC complex itself. So what we have observed is that the ORC complex is made up of 1, 4, 5, 2, 3, 6. 6 different proteins, and therefore a heterohexamer. Now let us look at how this origin replication complex is formed. Because it is a complex, again there would be a way by which it is assembled. So what is observed, especially in the humans, that is the Homo sapiens, and also in the Saccharomyces cerevisiae, and several other species have also been studied. And what has been observed is that more or less the way the ORC assembles is quite similar. So it is observed that you have the ORC2 and the ORC3 first forming the interaction. So the first interaction while making the entire complex is between the ORC3 and the ORC2. This complex, the dimerization of the ORC2-3 is enabling recruitment of ORC5. Once ORC5 comes in, ORC5 enables recruitment of ORC4 and ORC1. Keep in mind that all the three of these are AAA plus ATPase superfamily. They belong to the AAA plus ATPase. So they actually have a lot of structural similarity or homology. The domains that they have are very identical. And to this complex of the 5 ORCs can come and bind the 6th ORC, which is ORC6. So together, this entire ORC6 assembles to kind of form a structure like this. It resembles what we know as a cashew nut. Now this cashew nut structure, as you can see, has a gap over here where it can hold the DNA. So effectively what one observes is that the ORC complex can encircle the double-stranded DNA, and the several amino acids present in different folds of the ORCs are able to interact not just with the sugar phosphate backbone but also with several nitrogen bases present within. Interestingly, ORC1 can interact with the highly conserved T regions of the A element in the Saccharomyces cerevisiae. So there is a domain present which has amino acids that are conserved, and those amino acids can directly bind to the T's present in the A element. What is also observed is that the ORC4, in not just in Saccharomyces cerevisiae, but also in the Schizosaccharomyces pombe, has a very species-specific helix, and this species-specific helix in certain species is what is called as an A loop, and what has been observed is that that loop is able to bind again to the DNA very effectively. So in Schizosaccharomyces pombe, in fact, the interaction of the ORC with the DNA is through ORC4. While in case of Saccharomyces cerevisiae, that is Baker's yeast, the interaction is by all the 5 ORCs, 1, 4, 5, 3, and 2. The ORC1 through the minor grooves, the ORC4 through the major groove, and the 2 and 3 also through the major groove, but these bind to the B1 element, while these bind to the A element. So this is something that has been studied extensively. One important fact when you look at the structure of the ORC is that they share certain domains which are similar. So what is observed is that each of the ORC has 3 domains. One that is called as the REC fold, that is one domain. So this domain has a structure very similar to the REC protein. So therefore it is called as the REC-A fold. So the motif present is very similar to that present in the REC-A, and one knows that the REC-A is able to bind to double-stranded DNA. Thereby, probably this fold has a role to play in the binding of the ORC to DNA. But it also has what is called as a winged helix domain. So there is an alpha helix present in this, and this helix that is present in the winged helix domain is able to interact with DNA, while there is also what is called as an alpha helical lid. So all the ORCs have these domains present. When you observe each of the ORC, you will find that they have these 3 domains definitely present. The ORC1, interestingly, at its end terminal has another domain which is called as the BAH domain, bromo adjacent helix domain. Now, this domain allows the ORC1 to interact with nucleosomes. Well, the understanding is that the DNA in most eukaryotes are present in the form of chromatin. So, for the ORC complex to interact with DNA, its binding to the nucleosome will help the ORC to bind to the DNA. And that binding, initiating the binding of the ORC to the chromatin is because of the BAH domain that is present in the ORC1. So therefore, that is an extremely important domain that is present in the ORC1. So it assists in what is called as the nucleosome binding, in the nucleosome binding. And the WHT that is present in most of the ORCs, including that in the ORC1, is able to interact with the DNA. So you have the BAH domain that can interact with the nucleosome, while you have the WHT domain that can bind to the DNA. So you have both happening together. When you look at the Saccharomyces cerevisiae ORC, you can see the structural similarity. You have the ORC1, ORC4, ORC5, ORC3, ORC2 coming together to encircle the DNA. So you can have the DNA being encircled here. So this is something that is important to note. One another important thing that is to be noted is that the individual subunits coming together to assemble with each other, that assembly is assisted by ATP binding and hydrolysis. And we know that the ORC1, 4 and 5 have inherent ATPase activity. Similarly, 3 and 4 also have inherent ATPase activity. So therefore, that ATPase activity allows the 5 components of the ORC to come together. In Drosophila, in fact, 5 ORCs are present. There are in that case you don't have the ORC6 present. So you may have slight differences in the ORC complexes from species to species. Here, what is important to be noted is that ORC1 can bind to the ATP, but the ATP binding of the ORC1 is not important for binding to DNA. However, the ATP binding domain of the ORC4 and 5, when mutated, then the pre-RC complex formation has been affected. So you will not have the ORC formation itself if the ATP binding domain of the ORC4 and 5 is mutated. So which means that ATP binding and its hydrolysis is important for assembly of the complex itself. What is also important is when ORC4 binds to ORC1, there is a specific loop with an arginine present that inserts itself into ORC1. Only when that specific arginine inserts itself into ORC1, the ORC1 ATPase activity is initiated. So therefore, such interactions allows the ATPase activity of the ORC complexes to be initiated, and they have a lot of regulatory and coordinated functioning, structure function aspects included. So that is important to note.